Okay, well, thank you, everybody, for great turnout today, both online as well as in person. It's a pleasure to introduce one of our newest faculty members, this other newest faculty member sitting right here. Gustavo started in September?
Yeah.
Yes. September. He's got his lab downstairs called Move-In.
Yeah.
It's kind of funky. He's coming to us from Toronto, where I spent a lot of time as well. He was actually at the Toronto Rehab as well as the Kreml for a bit. And his research really spans the spectrum from animal work into human translation and robotics. So it's a nice package, and I'm looking forward to his presentation on movement, neurorehabilitation, and neurorepair.
Thanks very much, Randy. Thanks for the invite. Thanks for Kelly for organizing this. It's an honor to me being here and presenting this. It's a lot of fun as well to talk about my lab. I have been working a lot on some grants to get some funding to hire some personnel, and it's exciting to show you some of the work I've done in the past. First, we are here in the First Nations territory, and we're very honored to be here as well. And then just want to talk about myself, my ethics in research. As Randy was mentioning, have a long journey of working with animals, but I also have been working with patients, and I really think it's very important for us to have this type of work, working on both ends. So what I see in the field, many researchers, they only do animal research, many researchers only do clinical research, and they don't like each other. For instance, I asked, they don't get along, they do not understand each other very much. So I think it's very important that I have the both experience, right? And and I have been working with animal models and surgical procedures, from surgical procedures to computational approaches, softwares, and then the whole package. So it's really something that makes me very happy with where I am today, and with all the work we have been doing. And I want you guys to emphasize with the work we do. The work we do is not for us, it's not for for anything else than the patients, right? You're thinking about them, right? I know any of you here, you may have a parent, some family, or someone that has a stroke or a spinal cord injury, and they need the answer, they need the cure, they need it right now. So we are working hard, and we're doing this for them. So I really want you to emphasize, and then I've used animals in my research, but it's for a good reason, right? We really are not there yet. We do not have a cure for spinal cord injury, for example. We do not have a cure for severe strokes, for example. What we do, it's all about movement, rehab, and repair, right? So movement, it's all about the movement. My lab is all about the movement. We are, using computer vision and other techniques to quantify movements in rats, right? Rehab, we do rehab in animals, right? And I will talk about why it's important to do rehab in animals. We have, preclinical studies of rehab in animals. It's super important, right? I will talk about this during the talk. And repair, it's we're looking, for combinations where we can combine rehab with other things. I have been combining rehab with stem cells, for example. But we have many other things that we can combine, like different drugs, antibodies that boost and try to act in conjunction with rehab to improve function for patients. And all of those treatments, they must be understood in the animal ecosystem before we can translate it to the humans. So it's all about the humans, the movement. And Einstein was talking with his kid, they say in 1930s, was saying to his kid, teaching his kid to ride a bike, and he was saying, life is just like riding a bike. To keep your balance, you need to keep moving. It's so real when we decide not to move. For example, we take a less active lifestyle. We really start to feel the consequences as we age, right? You can feel the consequence of having a sedentary type of life. Same when someone loses the ability to move after a stroke or after a spinal cord injury, then the individuals can really feel the consequences of not being able to move. And I think it's really interesting to think about rehab, because rehab is all about the movement again. If you go to the rehab clinic, look at the patients, The physiotherapists are making the patients move, right? Using robots or using electrical stimulation, and by moving you can regain movement. So I was talking about myself, and then I'm not the C-squirt, okay? It's about this story is about myself, but I come from a background of electrical engineering, and then I transitioned to kinesiology, so I was a student just like many of you here, kinesiology student, and I transitioned to master's in neuroscience, and then my professor at that time was a mechanical engineer, right? And his line of research was a new professor in the neuroscience department. His line of research in the neuroscience department was all about force plates, accelerometers, kinematics. And I was there. And then people at the neuroscience department, they were like giving him like, what does this have to do with neuroscience, right? People were studying dreams, things like higher level things. Just like movement was something secondary. Like you just move, there's no problem. So what does this have to do with neuroscience? And I was very touched by that, and I got acquainted with Daniel Wolpert and his TED Talk and his books and his articles. And one of his TED Talks, he talks about the C-squirt. Okay? So this is a story about the C-squirt. So the C-squirt is a little creature. The C-squirt is swimming in the ocean. So the C-squirt is swimming in the ocean. Swimming in the ocean. And for swimming, the C-squirt is using a very primitive central nervous system, okay, for swimming. But one day the C-squirt, she finds a rock, and I don't know why, the C-squirt will attach to that rock and will stay on that rock for the rest of its life, okay? Don't ask me why. Maybe the rent of that rock is very good. You know, the rent is crazy around here in Vancouver. I don't know, if I find a good rent around here, I will attach to that rent, I will never do it again, okay? The C-squirt attached to that rock. The first thing the C-squirt does, it digests its own central nervous system because it doesn't need to move anymore. Yeah, they do that. Yeah, you look surprised. Yeah, they do that. And why? Because it doesn't need to move anymore, right? So why does it need the central? And actually, the only way of us interacting with the world around us rather than moving, is by sweating. So this is how important our brains are for movement. And we are not the C-squirt, we are very complex creatures, right? Around 4 to 10 million years ago, our ancestors decided to assume this bipedal posture, and they could free their hands. And with their hands free, they could do all sorts of movements using their hands, like building tools, and later on writing and things like that. And with this, it began the most striking part of human evolution, right? Our brains expanded, and now we are humans, and we use a lot, the hands a lot, right? So we are not the C-squirt. We have 86 billion neurons. We are very complex, and I like this number because it comes from I'm Brazilian by the way, a Brazilian neuroscientist, Susana Herculano Rosell. She's very famous for making brain soup. She takes the brain, makes a soup, and then she counts every cell. She counts the number. So we thought we had like 100 billion neurons. So Susanna came out with this number, 86 billion neurons. So we are very complex. We are controlling more than 600 muscles. We have more than 20 muscles in your hands, right? And 39 if you count the extrinsic muscles as well. We are very complex creatures. As a consequence, our cortex expanded, and I'm showing here all the gyros and sulcus. So gyros are the proeminences, and sulcus are the valleys of the brain. And of importance for the talk today is the precentral gyros, right? Here in red. Oh, that's cool. Okay. In red. So in this part of the brain, most of our neurons controlling the movements are located, right? The motor region of the brain. And then Wilder Penfield, like you see on this graph here, he did mapping of those regions. So Wilder Penfield, a Canadian neuroscientist, he would lower electrodes and would stimulate each of those regions here and would observe the movements that the patient would do. So for example, it would stimulate here and the hand would move, right? And in the homunculus of Penfield, what we have, The size of each part of the body is, how big the motor cortex representation is for that part of the body. So what we see here, because we are humans, we evolved using our hands. The region controlling the hands is huge, right? So this is important for this talk today, because we are going to talk about how we use our hands and how it's important for us to have the connection from these hands to the spinal cord and to the muscles, and how this is a biomarker for rehab, right? So what I did in my first experiment for my PhD, I did something similar. So this is a rodent brain, so I lowered electrodes into the brain. I would simulate different regions of the brain with my colleagues, and we would observe the regions of the rat, the limb of the rat that would move, and basically the green dots here are when we saw paw movement. So in the rodent, it's very similar to humans, right? We have a huge region of the brain controlling for the hand. So now rehab. So my PhD was about rehab in rodents. So we had a stroke lesion in the rodents, and then people ask me, do you wait for the rats to have a stroke? No, we don't wait for the rats to have a stroke. We induce a stroke. So and we talk about my lab on the go, right? So this stroke was induced by endothelin 1. It's a very potent vasoconstrictor. It's a peptide that will be injected in the brain. It will make those vessels to constrict, and then there's an interruption of the blood flow, right? In my lab, we will be using a different type of model. It's called the Photothrombotic model, right? We'll talk more about this model later. We induce the stroke and we give the animals rehab. So some animals will have rehab, other animals will not have the rehab. And what we did see, first let's see the stroke. It's a very large stroke. You can see it's very large stroke. You have like a very pronounced loss of the cortex and also the striatum here. You see, between animals, the lesion was very similar. So the standard is the animal that did not have rehab, and ER is the animals that had rehab. So it's very important for us, and then we will be doing this in the lab as well, for us to control the size of the lesion, because you can imagine, most of the variants that we see in those models are from the lesion. And what we see with rehab, both in the clinic and in the animal models, the variance with rehab is very small. So it's a huge confounding factor, and it would be amazing, you guys would be amazed to notice, to appreciate how many labs in the world, including some labs I was working, big labs, they're not worrying about it. They are not worried. They don't have a clue of what's happening with the lesion. This is the most important. So our lab, we are a humble lab. We're starting slow, but this is the basic well done, right? This is the movement rehabilitation, the repair lab. So it's very, very severe lesion. You see like the contralesional cortex, the intact cortex and compared with the affected cortex, there was a huge loss of cells. But what we did see is that animals that had rehab here in panel D, so they are the field circles. We did this staining, so around the lesion we did this staining for FOSB. FOSB is a marker of chronic use-dependent activity of cells. So basically, if a cell is super active, and they use this for addiction, right, in rodents, they would see, reward systems would shine with this type of staining. We also use for rehab, because rehab is something that you're chronically using other regions of the brain to compensate the loss of some of the regions, right? And what we did see is that rehab induced this more pronounced use-dependent activity around the motor cortex of the animals. It's not only that, it's not just that. The regions are more active, but also they're more functional, right? So with rehab, with the repeating of the movements, the regions around the cortex, the lesions, so the lesions are those circles here, and then here the maps, as I showed before. The blue maps are the movements of the proximal limb, and the green maps are the movements of the hand. We can see that animals that had rehab, they undergo a type of plasticity, where regions of the brain are more likely to become responsible for the control of the trained part of the body. So we are training the hand, we have more regions controlling the hand and the rehab animals. So the cells around the lesion, they are more active and they are undergoing this plasticity. So rehab, this is the mechanism of rehab, right? So rehab is acting like this in the brain. And this is how patients can recover after stroke, for example. So we can see here, as I was saying, that the size of the lesion, so the greater the size of the lesion, the smaller is the area, residual area, it makes sense. And also, animals that had rehab, they had more expansion of regions specific to the train part. Just to explain the graph, right? But it's just repeating what I said before. And how we think this is happening. So this is something that we are studying in the lab. How we think the mechanisms of rehab are happening in the brain. So when we understand the mechanisms, we can also understand how to improve it, right? And so this is some studies from Castro and Amancos. So we have these loops, right? So the thalamus sends information to the cortex will send information to the thalamus, and the thalamus will send information to the cortex can send information to the stratum will send information back. So creating these loops. So what Castro and Amancos did, he implanted electrodes into the thalamus, right? The thalamus right here. And then Castro and Amancos would be recording the neurons, the activation of neurons in the thalamus as the animals were learning a new skill. So the animals are learning. So he would see the sequence of firing, the pattern of firing, the oscillations of firing. And then after the animal learned the skill, the animal was returned to the home cage and was left there. So the animal would eventually go to sleep. And we know that sleep is a very important part of consolidating our memories. So during the night we resonate, we reverberate the memories, and we can consolidate the memories. It's very well known in the hippocampus, for example. The thalamus does the same thing. During sleep, the thalamus is recapitulating the activation with the same pattern of activation as seen before, such as consolidate the skill that was learned. So this is how we think that rehab is acting to promote recovery after a spinal cord injury. After stroke, sorry.
Question on the previous.
Yeah.
Just two quick questions.
Yeah.
I'm curious, first of all, whether the involvement of striatum had impact on the recovery. So I think since you do have variation in the size of the lesion, were there some animals that did not show any striatal lesions?
Yeah, no, good question. No, we injected striatums on.
Yeah.
Yeah, we had 2 injections through the cortex and 1.
Okay, and the second question then from that is there is variation of lesion size, right? And I was wondering, since you have controlled injections, is it a vascular variation that's causing the variation in lesion size between rats?
Yeah, good question again. Yeah, the injection procedure, sometimes you have spillover of the peptide. Okay. Many things can happen. Sometimes you think you're deep, you're not that deep. Many things can happen. But what we did, we did a good job. We had MRIs. Had MRIs. And here's Paul, the MRI guy. We have rat MRI. So after the lesion, we would send the animals to the rat MRI.
Yeah.
We would see the site, and then we would, split the groups
by size.
Evenly. Yeah.
Okay.
Exactly.
Okay, thanks.
Yeah, thanks for this break. I was needing some water. Okay, so the first part of the talk was the motor cortex. So, okay, the motor cortex, and here you like this, Randy. I like to talk about the motor cortex as the maestro. So the motor cortex is very important. You guys did see how it's important. It's related to the function, and when we rehab, you have it's the expansion of the motor cortex that supports recovery of function. And the motor cortex is really controlling movement, right? Everything is generated, excluding reflexes, of course, but volitional drive is generated by the motor cortex. And I like to think of the motor cortex as the maestro. So we are motor cortex is saying go, stop, things like that. And the lower motor neurons in the spinal cord, they are like the musicians. They are listening to the maestro. And don't get me wrong, the lower motor neurons, they are very good. The musicians are very good as well. Musicians, they train for the whole life. They know how to play their instruments. They are super good. They are ready to play. But if the maestro isn't there to say, hey, go, stop, they don't know what to do, right? So this is something that happens after a spinal cord injury. So after a spinal cord injury, you have the disruption of the commands from the motor cortex to the lower motor neurons. So from the maestro to the musicians. And what we see is like weakness. Patients, they cannot move, right? So patients try to move, so the maestro is telling for the musician play, but the musician cannot see the maestro, so it doesn't start, right? So there's no movement, or the movement is weak, or you have lack of coordination between muscles. You have, events, different frequency bands. You can have involuntary activation. You can have muscle spasms and things like that. So it's not only the motor cortex. So the first part of the talk was about the motor cortex, but no, we evolved. We are humans. We use our brains for controlling the hands, but we need a connection from the brain to the hands. When this is disrupted, it's a very serious problem, right? And we did test this in patients living with spinal cord injury, and we were using TMS, transcranial magnetic stimulation, to stimulate the motor cortex, and we were measuring the response on the hands, right? We did other types of assessments as well. We had the integrity of the somatosensory pathways. We had the integrity of the lower motor neurons. I will not focus on that because I don't have much time. Oh, I didn't start my... Oh. Okay. Okay, I don't have much time. So let's focus on the red, right? So the MEP, so testing the connection from the brain to the hands. And we had assessments very early after the spinal cord injury. 4 weeks after the spinal cord injury, we would test this integrity, and we would use this as predictors in a machine learning model, right? So we had other types of variables as well. We had clinical variables that are related to, the strength of the limbs, the sensation of the limbs, and things like that. But we also had this integrity of the brain to hand projections. And the machine learning model is trying to learn and predict based on information. So basically we had the clinical information, the machine would learn and would try to predict. Now we give another book to the machine. Now you have the connections from the brain to the hands. Learn that. The machine will learn again. And what we did see, forget about this, it's a lot, but just this graph here. The model where the machine has the book telling the machine, I'm getting creative right now, sorry guys. So we had this information about the brain-to-hand projections could predict better. So how do we know that? The area under the curve of the dark model is when the machine learned without the information of the brain-to-hand projections. And the red model is the model when the machine had the information about the brain-to-hand projections. And what we see in the y-axis is the ability of the machine learning algorithm to predict the true positive class. The true positive class is I'm trying to predict if the hand is going to recover, and I predict yes, it recovered. This is a true positive. Positive is defined because recovery I define as positive. It's true because it got it right? And the x-axis is predicting the ability of the machine of predicting the false positive class. So the false positive class is something that we are very concerned, because if you're trying to predict someone is going to get Parkinson's, for example, you don't want to predict that you're going to have Parkinson's and you will not have. This is a false positive. So people are always trying to have a good metric here. So what we see here is that we have both metrics are improved. You can see that it's a bit to the left, a bit up. So the machine learning model could predict better if the person would regain the strength of the hands 1 year after the spinal cord injury. Right? So we are measuring 4 weeks after the spinal cord injury. I want you to have empathy again. Imagine if you have a spinal cord injury, what's the first thing you ask the doctor? Will I get better? Right? So 4 weeks, and people, they lose, there's something called spinal shock. People lose, they are flaccid, but because of spinal shock, and they can regain that, right? So do you want, if you're saying to someone you're not going to recover, you really want to have better predictions, right? And then we are showing that we can have a better prediction because many patients, they are classified as AGIA. AGIA is the most severe type of lesion, right? And the doctors say, yeah, you're AGIA, you never walk again. And around 20% of those patients, they're walking, right? So it's a burden for them, right? So, you can imagine, right? It's a very tough moment. So our models are trying to help, and then we are interacting with people, researchers in Europe. We are now approaching ICORD as well. We want to further develop those models. And Martin Schubert, he called me this weekend. He was asking for, we were working with Electrophysiology, trying to know what we are measuring now, what we can do better using Electrophysiology because we think it's a good predictor for spinal cord injury. And then we can see like having this biomarker. So biomarker, and when I say biomarker is the integrity from the brain to the spinal cord, we see the MEP here. Patients that did not have the biomarker early after spinal cord injury, the baseline motor score was 0. So baseline motor score 0 is I cannot move, right? And then 1 year after, here's the change in motor score. 1 year after, patients still couldn't move. But if the patients had the biomarker early after the spinal cord injury, and you can see that early after the spinal cord injury, the motor score was 0 for most of them. They couldn't move, similarly, right? But 1 year after the spinal cord injury, they could improve the strength. Many patients reaching a score of 5 is complete movement of the hand, right? And then we see that the greater the integrity of these brain-to-hand projections, the greater are the likelihoods of recovery in the yellow. And then plasticity as well, we'll talk more about plasticity later. With time, patients are regaining strength 1 year through the course of the recovery. The integrity of the brain-to-hand projections is increasing, right? So some plasticity is happening there. We are using this for prediction as well in another study. So here I have a patient. This is electromyography. So I asked my patient to do this. This patient couldn't do anything, couldn't move. But in the electromyography, I could see some activity, right? You see some motor units are firing. 40 sections of therapy later, you could see this patient was moving, right? Was moving. So we are using this for predicting the response of this therapy. It's called functional electrical stimulation therapy. So basically when a patient comes to the clinic now before study, we can test the different muscles of the patient and we can say, hey, this and that muscle, we can see something. Let's focus the therapy on those muscles, right? So we have, the therapy time is very limited, it's very expensive. So therapists, they are trying to find better ways of optimizing rehabilitation. We call this precision rehabilitation, right? So the integrity of the corticospinal tract is very important, right? So why? Why it's so important? There must be a reason, right? Must be a reason why it's so important. And this, Randy will like this one as well. It's a very recent paper. So evolution. Novel regions are expanding in our brain, including the precentral gyrus I was telling before, where the motor neurons are, right? So this is number 4 in the graphs, number 8, sorry. But especially the prefrontal cortex, right? So those regions, they were comparing MRI templates between monkeys, non-human primates, and humans, comparing the differences to see where the brain has expanded the most. So they found out that regions like the prefrontal cortex expanded a lot, right? Which compares species. But with aging, so this is the T statistics for aging. So they had samples aged and samples not aged. They could see that regions that expanded more, they also lose more gray matter, right? And we're including here the prefrontal cortex, the precentral gyrus, right? Number 8, we see number 8 here. So it loses a lot of gray matter with aging, right? So because those regions that are novel, they also suffer more, right? It's not only that, So this is brand new, this is a preprint stage. This has been cited by our colleague here from UBC, Brad Hilton. And then it was published in the Nature Reviews Neuroscience a few weeks ago, and he's citing a preprint. So this work is that important, right? For Nature, they allowed him to cite this paper. They created a new type of lesion. So basically, a mice is born, a very young mice, it's like a mice, newborn mice like this, and they are inserting a micropipette, those that you use for patch clamp, for example, the very small ones, and they created this new ultrasound type of lesion. So they inserted the micropipette into the corticospinal tract, the brain-to-hand projections, and then ultrasound. They can create now a very small lesion in the corticospinal tract, the brain-to-hand projection, right? And they did it at different time points, right? So this is super interesting because they show that when you do the lesion at P1, so 1 day after birth, so what happens with the corticospinal tract? It's growing. It's growing. You see a baby's like that, they move because it's developing, right? Right? So the corticospinal tract, both in humans and in rodents, you are born, it's not elongated yet. It's growing, it's still growing. In the mice, it takes up to 35 days to grow. So it's growing, and then if it's a P1, 1 day after the mice is born, they create a lesion right here, right, at C3, the cervical level 3. They create a lesion here because the axon is growing, the axial growth cone is growing, and they make the lesion close to the growth zone, right? When they do that at P1, it regrows again. It looks just like that. You have to believe me, I cannot show the full article here, it has 50 days. It will regrow just like that. But if they wait until day 4 after birth, so the axon is growing, so the growth cone is down below, they make the same lesion at C3, happens. They wait 35 days, nothing. So it's fixed. After this first growth, it's fixed. It doesn't grow anymore. This is why it's so important, right? So what happens, the corticospinal tract, the axons are growing, and then spreading connections everywhere. When the connections reach the cells, the gray matter of the spinal cord, signals tell, stop, you're good. Now all the synaptic pruning process begins, right? Because at development we spread axons everywhere, then we start pruning, right? And then it's fixed, cannot grow anymore. This is why we need the bridge, we need the corticospinal tract integrity, we need something here. If you don't have something, it may never grow again. It's fixed at development. So here in this side they are showing that at P4, if you do the lesion at T1, T11 it doesn't happen, right? You have regrowth because T11 is way below, so C3 is here, T11 is here, so at P4 the axons are still growing there, right? So this is very interesting because it's now we can learn more about machinery making those long track projections, right? So this is the integrity. So this is the work we are doing, and then so those are predictions in stroke. So work has been published in the Lancet Neurology and has many other publications showing the proportional recovery that we see when we have the integrity, right? If you have the integrity, you will regain strength, right? It's just like in this proportional, right? When I say proportional, people do not understand what's proportional. I'm trying to improve that, but let's say you have a flu, right? You have a very, very bad flu. You will get very bad, right? But when you will recover a lot, right? Right? It's proportional. You lose a lot, you gain a lot, right? And this, if it was like that, would be perfect for rehab. So people would be having spinal, I had spinal cord injury last week, but I'm good, right? People would be like that, but it doesn't happen. What we are trying to understand, what are those guys that do not fit that rule? What's happening? What we know right now, they lack the corticospinal tract integrity. See MEP positive and negative, it's corticospinal tract integrity. Patients that do not have it, do not follow that rule. They will have a severe impairment, will not recover. That data from stroke, this is our data from spinal cord injury, bit more dirty, but you can still see that individuals that do not have integrity, they tend to not regain strength. So we are replicating this now in our experiments in the lab. So we have the electrophysiological assessment and we can test the bridge. So we need the bridge. It's a predictor for for rehab, if rehab is going to work or not. And then we have to find ways. I really believe that we have to find ways to improve the plasticity of the corticospinal tract. This is the best shot that we have in our lifetimes to get those people to get a better recovery. And now we have all sorts of drugs and all sorts of things. But the good news about our lab is that I think, and many people think, that any drug, anything that you have, you will need rehab combined, right? So we are at that point. So we are seeing that clinical trials in stem cells for spinal cord injury, very good results, you know, on the dish, cell culture, everything's working fine. Go to the clinic, it doesn't work, right? We think that there's a missing piece there. We need the rehab, the targeted movement to direct the plasticity, right? How can we use the bridge to optimize rehab? So the long tract projections will not regenerate. I show you guys it's fixed during development. So we need the bridge. If we have a bridge, we can have short distances, plasticity throughout the spinal cord, for example, for shorter distances. For example, here we have a monkey, and they do an hemisection of the spinal cord. When you have the spinal cord here, you cut half of the spinal cord. So imagine spinal cord is going here. So here spinal cord is going here. This was cut here. So the integrity of the brain to hand projection was cut. You see no. So the darker parts here are the corticospinal tract projection, right? You see nothing here, right? But the other side, because it's an hemisection, the other side of the spinal cord has the corticospinal tract projection, so has the bridge, right? So you have the bridge there. This bridge, with time, so this is the intact spinal cord, this is the short term, and with time you see it's expanding. So with time, corticospinal tract projections from this side of the spinal cord can undergo plasticity and reach the other side of the spinal cord and re-innervate the de-innervated spinal cord. So short distance plasticity we are able to do. It's happening naturally. So this is naturally. So, and then we have the opportunity to enhance it with drugs, antibodies, stem cells, and try to build on this plasticity with rehab and targeting the plasticity. So they show that you can have, yes, plasticity of corticospinal tract, short plasticity. You see short term and long term, it increases the number of axons, the caliber of the axons, and you can see like the synapses, you can see the asymmetrical synapses. When they are excitatory synapses, they are asymmetrical, they can see it in the microscope. You see they are making contacts with the spinal cord. So yes, we can do something with rehab. We can induce plasticity with rehab. So we have the bridge, we can induce plasticity in the residual corticospinal tract projections. And how can we optimize it? This is where rehab kicks in, right? After a stroke, for example, we have these waves, right? So it's a wave of it's upregulation of growth promoting factors, such as BDNF, neuromodulating, and other factors. So the system is trying to heal, right? To have this window of opportunity, and then we combine. So this is the rat time. It's in rat time, right? So it's like 30 days. In humans, we know that this happens for 6 months and beyond, right? But especially during the first 6 months. So during the first 6 months, we can use rehab in combination with this endogenous repair, signalings to try to improve. But with time, you see there's a reduction, and the brakes start to kick in. So the growth inhibiting factors, because the system starts to fix itself again, and then with time, you see those proteins and the receptors related to making the plasticity stop, such as the NOGO proteins and the NOGO receptors, and the CGRPs that are components from the extracellular matrix. We know that we have 4 components in the synapse. We have the presynaptic, the postsynaptic, the astrocyte, and we have the extracellular matrix around it. This is just a thing that is blocking, it's like stabilizing the system, right? So CGRPs, we have the chondroitinase ABC, something, it's an enzyme that we can administer to try to break those CGRPs, the extracellular matrix. So we can combine those therapies at the late stage, for example. And this is very important for rehab and for those combined therapies, because the stakeholders, people living with spinal cord injury, people living with stroke, they are often in the chronic stage. So funding agencies, they are looking for chronic therapies. So we need something combined. I'm drinking a Red Bull right now, and then The founder of Red Bull has a son. That his son, had a spinal cord injury riding his bike. Now they are funding research. He's a chronic survivor. They need therapies for chronic conditions. And then we really think that in those scenarios, you really need to combine rehab with something else, with stem cells, with anti-novel therapies, with chondroitinase ABC. So this is something we will be approaching in our lab, and for this we are seeking partnerships because we are very good in rehab. We are not yet. We will be very good in rehab. Now we want to attract people that have new compound. I know that Brett Hilton is working with some ideas for trying to reopen that long track projection, regeneration, and things like that. And I think it's very promising for us. So I tried to do my part during the second postdoc. So this is my study I participated, where we're using stem cells in combination with rehab. So we were using a very peculiar stem cells. They are engineered stem cells, and we were pairing that with rehab. So what's special about the stem cells? They express GDNF. GDNF is very important because the injured spinal cord is a hostile environment. So things are happening there. And when you inject a stem cell in there, because of those chemicals and things happening there, it's more likely that the stem cells will become an astrocyte rather than a neuron. When you transplant a stem cell, you want the stem cells to become a neuron, right? So, and this stem cell is expressing GDNF because our lab, my previous lab, discovered that GDNF through the notch signaling, it's a long story, but through this signaling, it can, optimize the fate of the stem cells, so the cells are more likely to become neurons. And the stem cells are very interesting as well because they're expressing TTX. TTX is a very potent sodium channel blocker, expressing TTX under the doxycycline promoter. So when we gave the animals doxycycline in the shop, would activate this promoter, express TTX, and TTX would block the activation of the cells. So we could erase the cells, right? And we did measure the brain-to-hand projections using the MEP, right? We would stimulate the motor cortex, record on the hand of the rat, and we'd see the response. So this is the response after the animals had the rehab, they had the stem cells, you see a lot of response there. Then we give the animals doxycycline, you see that the response, the cells were deactivated and the stem cells deactivated and the response was reduced, such as the stem cells were building up that bridge, the broken bridge. We're trying to help there, right? We're trying to reconnect the brain to the muscles. Rehab also was important because it increased the survival of the cells. Here is staying for Homer. Homer is a human antibody because the stem cells we are using in the rodents are from humans. So we could see only the stem cells. With rehab, more stem cells survive. And here's a FOX3 marker, a marker for neurons. We can see that animals that had the rehab had more of the stem cells becoming neurons. Okay guys, this is the lab. So this is my dry lab right now. We'll talk a little bit about the lab, the projects we're developing. The dry lab, we have the 3D printer, we have Arduinos and Raspberry Pis. So this is what I could do so far. I could remove things from the box and leave it there, but we are planning many projects where we will build, I will show you guys the automated rehab. Because rehab, it's not just like by coincidence that people are not doing rehab in animals. It's a lot of work. I've done that. You have to manually go there 4 times a day, put the rats in the thing, blah, blah, blah. You have to put forceps and it's a lot, it's a lot, it's a lot. So we are trying to automate that. I will show some of the automated rehab that we have and we're working on more of those, right? We are computer vision guys, we are doing kinematics, we already have lots of videos for analysis. We are working on the electrophysiology. Electrophysiology is how we measure the connection from the brain to the paw of the animal. And of course a lot of histology, paying attention to the lesion volume, doing the basics, doing it well done, automation, and also clinical analysis. Right now we are looking for partnerships with people that have, researchers that have data sets already acquired because we don't have a clinical lab. We will not have a clinical lab for a long time, but we can keep the work, and I will engage with having people with lived experiences. For grants, some grants are mandatory. The Department of Defense, we need a ladder for someone with a spinal cord injury, for example. So I want to really engage with patients. I don't want to lose that. So I'm working with Victoria Clayton to be associated with I-Cord, so I can have this clinical part of the work as well. But right now I'm working on a code where I can generate my own patient data. It's a synthetic patient data. I will show a little bit of that as well. So the first projects of the lab will be the lesions, the basics. So this is the photothrombotic lesion. It's a very interesting lesion because, for example, for the brain lesion, you don't have to open the brain of the animal. It's all done by light. So we inject a dye into the animal. It's a rose bangle dye. You go into the circulation, and then with a strong light, once light's strong enough, it goes through things, right? It can go through the skull of the animal, the bone, and then can penetrate the brain. And whenever, every place the light will illuminate the brain when the animal has this dye, will cause a reaction, an oxidative reaction, right? So we create a reactive oxygenic species, and will cause the plaquettes to agglomerate and cause like a thrombosis, right? Just like when someone eats too much of a McDonald's and have a heart attack. But it's not fat, it's like, conglomerates of plaquettes, right? And we cause the lesion, so it's a very interesting lesion. And surprisingly, I could find only 2 papers doing this in the spinal cord, right? So we are approaching this as a very cool project. We are developing the lesion models. And then for the spinal cord, it's a bit more complicated. When you're doing a surgery in the spinal cord, you have to go through three layers of muscle, open it, but you don't have to expose anything, just shine light on the spinal cord. So we are hoping it's going to be a good lesion for, in terms of, the health of the animals. And then we have to test that, something that's new. So this is the main thing of the lab, the lesion, and looking at the lesion. And of course, computer vision. So I got started with deep lab cut. So deep lab cut, basically you can do markerless tracking using computer vision. You have to train a network, and then the network with some time will learn what you're trying to see in the image, and then we load new videos, the network can track it automatically. So right now I have one rat before a spinal cord injury, so the movements of the hand, and then after the spinal cord injury. So we're really interested in understanding the movements of the hand, and if the therapy can really promote the recovery of the movements of the hands. Because what we know in people that work with behavior in animals, the behavior we do in animals is not very sensitive. Sometimes for Parkinson's you are looking how many times the animals, yeah, you can do better, we can do better than that. So that's the movement part of the lab, right? So we are here in Kinesiology department. This is a very strong part of the lab. We are working with movement. And this is something that I learned people need it. They really need it. And they think it's easy. It's not just like before I was talking about when I was starting, people they think it's easy. They try to do that, they cannot do it. They can't. It's complicated. And we are in the perfect place to work with movement. We are in the kinesiology department. And this is so important for novel treatments. You want to be sure when you say it's working, right? It's working, I know it, I'm sure. Because now we spend millions of dollars in a clinical trial to see what that doesn't work, right? This is very important. So we have some videos already collected. And I'm looking for people that want to have fast papers. It's all collected. We work in agreements with the University of Ottawa, so I collected those data. University of Ottawa, and then we can discuss this later. This is some sample of our electrophysiology we are doing in the lab. So basically, I implant an electrode. It's a screw electrode over the motor cortex, and I stimulate the motor cortex, and I can obtain responses. So this is our a recruitment curve from my electrophysiology. So I performed this experiment in 11 mice. Before I did a spinal cord injury, I call it here cortical spinal tract disruption, because I prepared this for a grant. I cannot say spinal cord injury. But it's a before spinal cord injury and after spinal cord injury. You can see that after the spinal cord injury, the projections from the brain to the hand, they're less exciting, right? And you can see here, before the spinal cord injury and after the spinal cord injury. And then you can see here, a healthy animal, we see the MEPs, and then the animal has a spinal cord injury, there's a delay of the latency, there's a reduction in the amplitude. So we've been doing this, a lot in the lab. It's similar to what we do. Here's myself doing a TMS, the CAMH in Toronto, the Center for Addiction and Mental Health. I did a course there last year. So it's something similar that we want to do in patients in the long run as well. So we just need more time, but right now this is what we're doing. And this is the automation part. So working a lot with GitHub repositories and the open behavior, they have everything there. So this paper is the first one I'm working on. So they have the 3D printed parts ready for printing. I will start printing this in a few weeks from now. Plexiglass parts. This is the equipment that you see in the labs. And when we enter the lab, the guy has one single equipment for 20 years. I wanted this one to be mine because it does everything. I had my previous supervisor working with memory. So I have fear conditioning memory. So he like electrocuted rats and like see the freezing of the rats. They had like a very old wooden box, like for 30 years, you know, standing in the lab, like had PNS, like big papers with that little thing. I'm planning to have something like that. It is so important for my lab because it has everything. It has a camera, and it has mirrors here, so you can have a 3D reconstruction of the movement. It has sensors that can sense where the animal is, and then it can trigger the cameras, right? And it can send TTL pulses to implants and can simulate the brain, and you can time things, you can understand what's happening in the brain. You can do everything with this box, right? So we will be working a lot on, you know, modifying, assembling those parts. They're working with Arduino Uno. This is an Arduino Uno. I have a few in my lab already. Microcontrollers, sensors, really a lot of fun. Just connecting things, follow the recipe, and troubleshoot. So this is something very interesting, and I'm planning to have a few master students, one for more of the behavioral part of this, another more for the rehab part. So people that are more interested in you know, electronics and computer programming, things like that. So it's a very interesting project for us. And the last thing I want to say is, they said I had 1 hour and a half. Okay, it's Kelly's fault. She told me 1 hour and a half. I was preparing 40 minutes. So you have 1 hour and a half. Super important, clinical trial design. Money. It's all about the money. It's so expensive. Now you're going to have a clinical trial. Now you want me to pay for rehab. People they don't want to do that. So why studying rehabbing animals is so important? Because we have to show people that it's important. It's super important. They have to plan ahead and have, in future clinical trials, have the combination rehab with rehab. It's not only any type of combination. When you are applying the rehab, is in parallel with the therapy or is it sequentially, right? What's the dose, right? So we need to know, and I will show one example, it's very brief, one I promise. This paper was published in Science, right? The anti-nogotherapy I was talking before. The nogotherapy, it's an antibody. They're trying to block the nogo protein that activates a receptor. This receptor blocks plasticity. So they're trying to block that and enhance plasticity. So what they did see in rats, when they applied the anti-nogo antibody therapy in combination with the rehab training, the rat group, this is the improvement in hand function. You had no improvement in hand function. And you had a lot of plasticity. You see, this is the corticospinal tract. So this is, the lesion was on this side of the brain. So you see that the other side, because it's crossed, right? The other side of the spinal cord, you see no corticospinal tract. But the intact corticospinal tract from the other side of the brain, you see here, the bridge was there, could extend axons from one side of the cord to the other side of the cord. And with the nogotherapy and training combined, you had the most significant amount of plasticity, but the least significant improvement in recovery. So not all plasticity is good, and not always rehab is good, right? In this scenario, if they had the therapy, the anti-nogotherapy first, stop the therapy, and then do rehab, then they could see a huge improvement in function. So this is, and they see that's less plasticity. So this is the synaptic pruning I was telling before. So you have a lot of sprouting everywhere, and then the rehab kicks in to make the pruning of the relevant synapses, right? So this is why rehab is so important, right? And we need to pay attention. This is why we're working on rehab in our, so this is the rehab apparatuses I was using in Toronto. We are working in collaboration with Keith Fenrich, who will be in the Society for Neuroscience. we build all this attachment to the enrichment cages, and then animals, they can enter the box, you see, and they can enter any time of the day, and they can do it just by themselves, and then they have, you know, it's trying to reach there, the pellet. We have photocells here that can tell us how many times the rats are reaching. Sorry. And animals are implanted with RFID tags, and then we have RFID readers on those platforms, so we know the number of the rat, which rat's doing what at what time of the day, and we can have the dose of the rehab. So this is the big thing in the lab. We are working very hard. We'll be working very hard in building similar systems for our lab. And the final thing that I'm also interested in is neuromodulation. So from my PhD, we did see the expansion of the motor cortex. I was talking before. We see the expansion, but we didn't see a correlation. Like, this is how much the animals improved the hand function, right? The y-axis. And here is the expansion that we see in the motor map. We are expecting that the bigger rehab, if rehab is expanding the motor map, the amount of expansion should correlate with the improvement in function, right? So the expansion of the map is supporting the function, right? And we did not see that. We see that the animals that did not have rehab, they had this expected relationship, right? But for animals that had rehab, they did not have it. Such as something else was explaining the variance that we see with rehab. And we and others, we are very focused on this structure here in the midbrain, the red nucleus. And then the red nucleus, we know that after a stroke, the red nucleus will receive much more inputs from the corticospinal tract, right? And then we know from my experiment that with rehab and after a stroke, the red nucleus will be much more active. And the red nucleus is a structure in the midbrain. It's a very, very interesting structure in the reticular formation close to the red nucleus. We see powerful Boolean-positive cells. Powerful Boolean-positive cells are inhibitory cells of the central nervous system. They usually contact the body of the cells. They are contacting the body of the cell in the red nucleus, providing inhibition. So there's a secret there. We want to understand what's happening, and for this, we will implant electrodes. So the final part of the lab, and I think it's a more ambitious part, we are going to be implanting electrodes in the brain and recording neuronal activity there, see what's happening with rehab, how things are changing, and then with that we can develop novel, just like for practice we have deep brain stimulation, right? And we know this works a lot. And then for rehab we can have some sort of similar device where after understanding the whole of those structures, the brain structures, we can have implanted electrodes on those and having the system working properly with electrical stimulation. And for this I'm providing those equipments, the electrophysiology for recording and for doing all those things in the brain. And this is a very fantastic equipment. It's a scan scope. This will allow us to make the full pictures of the spinal cord, full pictures of the brain, so we can count cells, we can see the corticospinal tract, we see the projections in terms of histology. Yeah, I was over here. That was it. It's just perfect.
Let's thank Gustavo again for the presentation.
Yeah, thanks.